And the next speaker is Idan Rosh from the University of Haifa. He will speak about a mutation in the IQSEC2 protein, which results in neuronal hyperexcitability of patient-derived neurons and deficient synaptic transmission. Idan?
Hi, my name is Idan. I'm a graduate student at the Stern's lab at the University of Haifa. Our lab specializes in precision disease modeling. In other words, in the creation of accurate models of disease through the differentiation of iPSCs. And in this particular project, I'm going to talk about how we examine the intrinsic properties of neurons derived from IQSEC2 mutation carriers. IQSEC2 mutation is, just a second, is a mutation which is closely associated in causing intellectual disability and is often accompanied by seizures and autistic-like features in almost of 25% of all intellectual disability cases. This particular mutation can be found on chromosome X, and although that is found on this chromosome, it escapes X inactivation, which means it can be found similarly in both sexes. The protein that is produced by this gene called guanine nucleotide exchange factor, which is a part of the NMDA complex that activates the ERF6, which has a crucial part in maintaining the ratio between inhibitory and excitatory receptors at the postsynapse. And especially in this case, the AMPA receptors through the regulation of the endocytosis of AMPA receptors. And as you can see in this illustration, the transformation of a GDP to GTP on ERF6 and its subsequent activation has a significant role in maintaining this balance. And as you can see in carriers of this mutation, there is a reduction in the density of the postsynapse of this certain type of receptors. So about this project, initially we used blood samples from mutation carriers, the IQSEC2 carriers, and those fibroblasts that were extracted went through transfection with Sendai virus containing the 4 Yamanaka factors, the SOX2, OCT3-4, KLF4, and the CMAC. After the creation of the iPSCs, we divided the groups by using CRISPR-Cas9 to correct the mutation, creating a valid control group. After we received the end product, which is the iPSCs, we use the protocol as you see before you through the administration of different growth factors at different time periods to navigate, to differentiate those cells to the end product, which is neurons of the dentate gyrus, granula neurons, both from this mutation and both from the control group. Our first discovery from the examining of the neural networks and the mapping of neuron types on those networks has revealed decrease in amount of GABA expressing neurons. Now, since this mutation is characterized by the appearance of recurrent epilepsy seizures, the decrease of those neurons is quite fitting and maybe can explain the mechanism that stands behind those seizures, those epileptic seizures. Further steps that we took, we measure through a wholesale patch clamp those intrinsic properties of those neurons at 5 weeks post differentiation. The result shows an increase in spikes, in action potentials, as well as in amplitude, and different of spike shapes between the two groups, as you see before you. And we've fastened after a hyperpotential, which means that the neurons can shoot faster than the other group, and with the narrowing of the spikes. RNA extraction analysis and sequencing revealed 361 differentially expressed genes. And the pathways that we found involved include neural differentiation, neural development, and exonogenesis, and the overexpression of potassium channels. Now, at 11 weeks post-differentiation, we noticed something really interesting. This hyperexcitability is a change with a hyperexcitability. We can see the difference in the number of spikes. We can see a lowering number of spikes with the amplitude and the shape. Gene expression that we took from 11 weeks showed more than 3,000 differentially expressed genes with more pathways, such as the transmission of nerve impulse, synaptic transmission, and neuron differentiation. At the different time points, we also noticed a reduction in synaptic activity as the neurons grow. As you can see before you in the different time points, you can see the lowering of events between the groups, and at the 11 week, you can see almost no events at the mutation group. And finally, a Western blot in immunohistochemistry, through the use of those methods, we discovered a reduced surface GLUA2 expression as the neurons age. And to summarize that, when those neurons that were differentiated from the mutation carriers, as when they are very young, we can see a strong hyperexcitability, which is being reduced by age. Also, we see reduced inhibition with the reduction in amount of GABA receptors, GABA neurons, which can explain the mechanisms behind the epilepsy. And those changes after, as I showed you at the last parts with the synaptic activity, may explain the problems with intellectual disability, which is associated with this mutation. And that's it.
Okay, thank you very much, Idan, for a fascinating lecture.